Okay, so I'm Brian McMorran. I'm a graduate student in Linda Baum's lab. And as you might have gotten a sense of from my presentation in the morning or her presentation in the afternoon, my project focuses on muscle glycosylation. And so the easy way to think about any protein glycosylation is to imagine a tree. If you think about the trunk that runs up vertically, that represents the protein backbone. And then all the branches and leaves that extend out to the side represent all the glycosylation that exists on any given protein. And just like how in nature, different trees grow in different locations, it's known that if you look at a muscle cell membrane, there's actually different glycosylation depending upon what location you look at. And when I started my project, if you think of the knowledge base regarding muscle glycosylation as a landscape portrait, there was very little known about muscle glycosylation. So maybe a tree here or a tree there. And so my project has worked very thoroughly to try and paint a fuller picture about where the trees are located and what their leaves look like, trying to get an assessment about what muscle glycosylation in mouse cells looks like. There's very little known about it, but there are some potential therapeutics that could come from it by manipulating the glycosylation of proteins or shifting trees that might grow over here with certain leaves to compensate when the trees over here are gone, to fill in empty voids in the landscape so we have a full picture. If you were to look at a picture regarding human muscle glycosylation, that would be a completely blank canvas. There's even less known about that. And so my project ultimately is going to transition to starting to work on painting that picture, looking what glycosylation might be there, and what leaves we should be focusing on if we're going to be working on translating glycosylation-based therapeutics into boys with DMD.
Very good. Okay, awesome. We'll cycle around this also. Are you ready?
I hope so. Hello, my name is Majib Jan. I'm in Dr. Pyle's lab. Need some music. We're basically trying to treat a very severe muscle wasting disease, Duchenne Muscular Dystrophy. In our lab, we're using a cell-based or a personal medication approach, where we're taking cells from the patient, cell biopsy or skin biopsy from the patient, and we're turning them into induced pluripotent stem cells. So these induced pluripotent stem cells, we're trying to develop a protocol for them to eventually turn them into myoblasts and eventually into full myofibers. And once that model is well developed in our lab, we can then use those cells to backtrack and get them to a progenitor or a skeletal muscle progenitor cell that we will essentially put back into the patient to not only fuse and correct the disorder in a cell-based manner, but it's also their own cells, so there's no need for a medication to immunosuppress them or so. So that is, in a nutshell, what we're doing in our lab.
Hello everyone, my name is Wesley Ulm. I am a postdoctoral fellow in Stan Nelson's lab. Our project today, I guess the best way to introduce this is you probably never thought you'd hear this in a million years or a billion years, but in a sense we're sort of taking a cue from the worlds of planetary and politics to help illuminate the molecular biology and potentially the treatment of Duchenne muscular dystrophy. Well, how is that? There's an important tool used in the fields of planetary and politics, the public opinion poll with a national sample. And in our case, we have a poll of our own, which we're calling Duchenne Connect, which is going to be the topic of what I'm going to present here. Except that thankfully, instead of getting public opinions, and thankfully also instead of calling people at 8:30 at night while everyone's eating dinner, getting everyone annoyed, we have a poll that gets facts reported by Duchenne patients and families themselves, and it's obtained via web access online. So Duchenne Connect is an assemblage of data on several axes in several areas from Duchenne Connect patients themselves, obtained from a nationwide sample. Now, why is this so important? Rare diseases in general are problematic because they are rare. And the rareness of them means that it's very difficult to get enough information to reach statistical significance. In practical terms, this means that a lot of the relations that we want to establish to get new information are very difficult to do in practice. Now, as a good sort of unifying principle for a lot of medical developments these days, they depend significantly on establishing meaningful relationships among many different factors. And oftentimes, those relationships are quite small. So we just need the power of numbers and economy of scale. We just need a lot going on, essentially, to amplify those relationships to see something meaningful. And Duchenne Connect allows us to do this. We're interested in several parameters, but in particular, intangible indications, i.e. indices, of patient improvement, such as an advancement in patients' age at loss of ambulation, i.e. at the point where they need a wheelchair. And we want to look at factors that may affect this. Not just sort of traditional treatments, but also things like insurance status, age of diagnosis, as well as, of particular interest, some remedies that you can potentially pick up at the drugstore, things like vitamin D, coenzyme Q. And we found, actually, from Duchenne Connect, from data that patients, their families, and also their treating centers have reported to us, that there are significant effects on the age loss of ambulation by these other factors. And we've also extended this a little further to even potentially find, to stratify and subclassify patients into groups based on a potential promising treatment called exon skipping, which is essentially sort of like resetting the Lego blocks together that make this gene up. So we've got a lot of potential and a lot more to look at in this data set, and there's a lot of potentially encouraging possibilities for applying it to improving patients' status. Thank you.
My name is Gibbs, and I'm a postdoc in Dr. Rochelle Crosby-Watson's lab. The focus of my project is in finding ways to better stabilize the cell membrane in Duchenne muscular dystrophy. As you've all heard today, Duchenne is caused by a loss of dystrophin, which is a protein that's normally found just under the cell membrane in muscle fibers and serves to protect the muscle fiber membrane during muscle contraction. Skeletal muscle undergoes a tremendous amount of mechanical stress on a day-to-day basis, and dystrophin acts as a shock absorber for this stress. I study a protein called sarcospan, which is found at the muscle cell membrane and interacts with a number of proteins that can directly compensate for the loss of dystrophin. We found that by increasing levels of sarcospan in mice with muscular dystrophy, we see a corresponding increase in membrane stability. These mice have better muscle function and less muscle damage. We found that sarcospan can alter some of the earliest events that occur in muscular dystrophy, and we believe that this makes it an excellent candidate for identifying new therapeutic approaches of DMD. Thank you.
My name is Haibin. I'm a postdoc with Dr. April Pyle. So the focus of our lab is to develop novel therapies for Duchenne muscular dystrophy. So DMD, as you all know, is caused by the loss of dystrophin protein in muscle fibers, which dystrophin normally does is to anchor the muscle cells to their surroundings so that they can remain stable during muscle contraction. However, without dystrophin, the muscles becomes very fragile and die very easily. So actually, in our body, there's a compensatory mechanism to counteract muscle death by activating muscle stem cells. So these stem cells, once activated, they start to proliferate and make new muscle fibers. However, in the case of DMD, because muscle fibers are dying constantly, the muscle stem cells are activated far more frequently than usual. And up to a point, they cannot keep up and become exhausted. And from then on, the muscle loss is kind of a one-way road, and over time, the patients lose their mobility and other vital functions. So therefore, we want to take a regenerative medicine approach by making the muscle stem cells in a dish and then transplant them back into patients to restore their muscles. So we're using human pluripotent stem cells, including the patient-derived iPS cells, because they can proliferate indefinitely in culture and they can turn into any cell types, including muscle cells. Also, because these cells are derived from the same patients that will later on receive the transplants, the chance of getting muscle immune rejection is extremely low. So what I'm doing is to guide the pluripotent cells towards a muscle stem cell fate by mimicking what's happening during normal muscle development. And also to drive our approach closer to a clinical application, I'm also planning to find out conditions that can expand the derived muscle stem cells so that we can generate enough amount of cells for transplantation. Since we are to supply various cells that can rebuild and maintain the whole musculature, it's very likely that even with a single transplantation, the patient is going to benefit in the long run or even for a lifetime. I truly believe that this is a very exciting and promising path toward a personalized treatment for Duchenne muscular dystrophy. Thank you.